Welcome back and excited for the further session, which are gonna as captivating like every other session conducted till now. Going ahead for the third session of the day, Dr. Urmila Aswar Madam, who is the chairperson with us. She has about she has about 80 years of academic and research experience. She has published 30 research publications in various peer-reviewed international journals. She also has completed 30 preclinical industrial studies and has made 60 national and international presentations. We welcome you, ma'am. Now I request madam to continue the session.
Thank you. Thank you, organizer, for giving me opportunity to uh, work as chairperson for this session. And I'm really happy to introduce Dr. Liji Sasidharan Madam. And it gives me immense pleasure to introduce her. She is working as pharmacovigilance scientist right now at QIP, UK. She has completed her B Farm in 2002 and M-Farm in 2005 from Tamil Nadu. She started her job as senior researcher, data monitor excellence Data Research Private Limited, Hyderabad from 2007 to 2008. She worked as senior healthcare professional assessor in pharmacovigilance department Satyam at Hyderabad, India. Then she joined at Ipsen Biopharm Limited, UK as pharmacovigilance data entry specialist. She then worked as safety specialist, PPD Global, Cambridge, UK, and then Silton, UK, and back with Ipsen Biopharm as regulatory compliance manager. She has also served as pharmacovigilance consultant at Glenmark Pharmaceuticals Limited, at GSK Limited, then Stockley Park and Allergan. Now she is right now working at QIP, UK. Her responsibilities includes she acts as a lead scientist for the responsible product within the oncology therapeutic area. She provides scientific expertise during signal detection activity. She is involved with scientific review and interpretation of safety data and authoring of aggregate reports such as periodic safety reports and risk management plan. She is involved with preparation and review of pharmacovigilance documentation relating to the safe use of products. She provides input for the preparation and review of IBs, that is investigative brochures, core data sheet, local label updates. She assists local safety officers in the implementation of additional risk minimization measures. She is involved with scientific review of safety components of marketing authorization applications. She is involved with the development of global risk management process for pharmacovigilance team as a part of globalization project and act as a subject matter expert. She worked cross-functionally to provide pharmacovigilance inputs. So with this brief introduction, I would like I welcome her to this program, and her title for today is Signal Detection in Pharmacovigilance. So I welcome her and I request her to initiate with her talk. Thank you.
Hello, everyone, and thank you, Dr. Mila, for your warm welcome and for my brief introduction on my experience and my background. Thank you very much. I don't need to talk anything more than this. Yes, I can directly go to the presentation. Maybe if you're okay, if I start sharing my screen and please let me know if you can see my screen.
Yes, ma'am.
Okay. So, as Dr. Urmila has mentioned, I'm within pharmacovigilance since a long time now, since 14 years. I've been working on different aspects of pharmacovigilance. and currently is working as a scientist in QIP, mainly on the signal detection part. So here, I thought I'll just give a brief introduction on everything, basic principles of signal detection. So before going to the signal detection part, the first important thing is the definitions of what is pharmacovigilance. I would assume that you would already know what is pharmacovigilance and all the basic definitions, but it's important to talk about this before we go to the actual signal detection part. So, as you might already know, pharmacovigilance is a science and activities relating to the detection, assessment, understanding, and prevention of AEs. It's not only AEs, but also any medicine related problems. So that's the whole definition of pharmacovigilance. The next one, what is an adverse event? The ICH guideline describes adverse event as any untoward medical occurrence. So it should be focusing on untoward medical occurrence. It only says that untoward medical occurrence, doesn't say that is related to the product. So there is no causality implied there. So it is anything that happens to a patient or a subject during a clinical trial investigation, anything that happens after taking a product. Doesn't mean that just it was caused by the product. So there is no causality there when you say adverse event. So the next definition here is adverse reaction. So that says a noxious and unintended response to the product at any dose. So there a causality is implied, means it's noxious and unintended response to any dose of a product. So that's the difference between the adverse event and an adverse reaction. So how do we confirm an adverse event as an adverse reaction? So that's the process in the signal management. Signal management. First, we need to know what is a signal. So when we have an adverse event and a drug, and we have a reported information on a possible causal relationship between the event and the drug, and the relationship is unknown or incompletely documented. So we research on that signal to make it clear or confirm whether it's an adverse reaction or not. Now, signal management is a process, is a cyclical process. This includes signal detection, it includes prioritization of the signal, evaluation of the signal, and finally, the action to be taken is a communication and risk minimization measures. It is a regulatory requirement for all marketing authorization holders to conduct signal detection or signal management. It's an important process within every pharmaceutical company. So you say why this is important. This is an integral part of pharmacovigilance practice. Why? To maintain the safety profile of the company's pharmaceutical and biological products. So every company, every product has got its own safety profiles, as in the labeling, ration leaflets, anything. So every drug has got its risk, and there is a safety profile of the product. And it is important for the company to maintain the safety profile, and signal detection or signal management is a part of it. To support the favorable benefit risk balance. So this is again throughout the life cycle of a product, benefit risk balance should be checked. If risk outweighs benefit, then there should be a definite action. So there's no point in taking the drug if the risk is more than a benefit. So the benefit risk balance should always be positive. And finally, to ensure the safe use of the drug by healthcare providers and their patients. So that's why this is important. And now let's see what you're looking for in a signal. A signal means something that occurred as a new unlabeled event. Unlabeled means which is not in the product characteristic or in the safety documentation. It's not in the safety profile. So that is unlabeled. unexpected change in the severity of a labeled event. So let's say there is infusion-related reaction is an expected event, but it has led to death means there is a change in the severity. So fatal infusion-related reaction is an unexpected change in the severity. So occurrence of an AE normally considered rare in the target population. For example, this is a product for pediatric population, and this kind of event is not seen normally in pediatric population, but normally seen in adult population. So then that's a signal that in pediatric population, this is an adverse event. Any new drug interaction with drug or food or anything else. And identification of novel at-risk population. Example, okay, this is something in a population of pregnant women or a population with underlying condition of renal hepatic failure or cardiovascular conditions. So at-risk population. So if there is a signal that shows that a certain population is at risk. Again, this is more the misuse or abuse of drugs, which is also common. Example is opioids, is misused or abused, addictions, anything. So any kind of drug can lead to a misuse or abuse. So that is a signal that we need to look for. So there are different sources of signals. The main one is ICSRs. ICSRs means individual case safety reports from the database. And these ICSRs from the database, they come from different sources. There may be regulatory authority reports. There are spontaneous reports directly reported to the MAH by consumers, patients, physicians, anyone. There are post-authorization studies conducted by the company. There are scientific literatures published. Then serious adverse events from clinical trials. This is also a source of information. And non-interventional studies like marketing projects, market research projects, etc. And one of the main or the recent sensitive source is the media. Anything about a patient adverse event might be reported in an internet or company website, journals, social media, anywhere. So they are also our source to get the information of individual case safety reports or patient adverse events. So we don't look only at adverse events. There are special situations we need to collect information on, is lack of efficacy, medication errors, off-license use, or overdose, misuse, abuse, as I said before, pregnancy, breastfeeding, lactation, pediatric use. So all these are special situations we collect in addition to any adverse events. So there are other sources for signals. So other than this global safety database from a company or these AEs and special situations, there are other sources for signals. That's other department data. For example, regulatory data requests. Sometimes regulatory agencies request for information because they have more information on a class of a drug. They might inquire. They might even suggest changes to the labels like SMPC, so summary of product characteristics. So they might suggest changes, so wherein we find it as a signal and we can also do further signal evaluation. Now, product quality compliance. So we get product quality compliance associated with adverse events. A device defect, anything can be a product complaint. Medical inquiries. Every pharmaceutical company has got their medical information department where a physician or a healthcare professional can inquire about safety of a product. And through the medical inquiries, we also get some kind of signals. Example is if you get a continuous inquiry of macular edema for a particular product, then that shows, okay, there is a lot of inquiries coming on this particular event for this product. So we need to look it further. So that's the source of signal as well. Again, another important source of signal is the aggregate data. Aggregate clinical trial data. This includes development safety update reports, which is prepared annually or according to the frequency. AEs, laboratory data, preclinical data, they are all looked or reviewed for any signals. Periodic safety update reports, or PBRR or PADR. In the US, it's called as PBRR. So these aggregate reports also are a source of signals. Then there may be milestone data specified in the risk management plan. So some products have for their risk management plan, they have effectiveness check, and based on those milestone data, we can look for signals. That's also a source. Again, not done. There are more sources for signals of a product. There are regulatory databases. MHRA in the UK has got IDAP. In the US, there is FAIRS. Other databases, WHO, WIGI database, WIGI-base, and registries, epidemiological data from registries. So these are all sources of signals. And there is also the product labels from other companies. So if there is a class of product, for example, fentanyl, there are other formulations with other companies. We can also look for their product labels from certain websites. From the Europe, EMC is a website where we can get the other product labels or SMPCs, and we can assess a signal to see if there is a class effect of an event. So there are different methods of signal detection. Every company has their own methods and processes, but these are the common methods: qualitative method and quantitative method. So most companies use both the methods. There is automated process, there are manual process depending on the size of the companies. Qualitative data is nothing but a manual review of individual case reports. Some of the companies use the designated medical events. So designated medical events is a list of designated events published in the EMA website. For example, febrile neutropenia is always considered serious and important. Acute kidney injury is another important one. So there are lists where we can follow, okay, if we have received a case of febrile neutropenia for this product, then irrespective of the number of events we received, we still look at in detail. So that's a manual review. Now, quantitative review. So quantitative review is done by statistical calculations of disproportionality or based on observed or expected ratio. So disproportionality can be calculated by fractional reporting ratio. It's like a statistical indicator. It compares the frequency between two different settings. For example, the number of events reported for this particular condition by a total number of events in the database. That's the statistical method of calculation. Based on observed or expected ratio, means observed ratio. For example, while in the clinical trial, this particular product showed headache 1 in 100. Whereas after 3 months, we can see, okay, now the product has come to the market. After 3 months, we can see 10 reports of a headache in 10,000 people. So the observed ratio was 1 in 100, but now we can see 10 in 10,000. So, okay, this has gone to a larger population. So 10 in 10,000 is expected ratio. So whereas if you have seen like 1,000 in 10,000, then the ratio would go up. Frequency of data review. So how often do we need to review the data? So this actually, it could be monthly review, it could be quarterly, annually, or it all is all based on the number of AEs or ADRs received in a year for that particular product. If there is any potential public health impact of that event, or and also depending on the maturity of the product, number of years on the market versus a new product which is just recently on the market. So the next step in signal detection is signal prioritization. Let's say when we reviewed the methods, the statistical method we have used, we found some signals. How do we prioritize and why we need this prioritization? Prioritization allows to take an action more expeditiously. So higher priority signals will be actioned faster if it has a significant impact on the public or the population, patients. It's going to be life-threatening condition or anything. So that will be prioritized immediately. Actions will be taken immediately. So signal evaluation strategies. So how do we evaluate the signal now? A systematical evaluation is required. So now we have the signals, we have prioritized the signal. How are we going to evaluate the signal? How do we determine if there is evidence, sufficient evidence of a causality? The first one is the METRA. So from our database, we should be taking the standardized METRA query to retrieve and review the similar cases. Standardized METRA query, I'm not sure if you're already aware of what it is, but METRA is a dictionary used in the safety database, and there are preferred terms and METRA queries, FNQs, which retrieve similar cases, similar preferred terms of events. The data may be further stratified according to age, gender, ethnicity, concomitant medication, or disease. So we can look at, okay, this particular event is shown on a particular age group or a particular gender, ethnicity, or concomitant medication and disease. What were the concurrent condition of the patient at the time of the event, or what were the other medications the patient was taking. So this will also show us, okay, this particular drug was taken along with another drug. It could be either of the drug or it could be a drug interaction that caused this particular event. So it's important that the data is stratified according to these. Now, a judgment. The final is the judgment whether its signal is verified, means a confirmed signal. It depends on the number and quality of the case reports. And I say quality of case reports, most of the time, the individual case safety reports will not be complete. They may be consumer reports, they just report that the patient experienced some event without additional information. So there may be limited information on a particular event. So quality of case reports, it always depends on that, and the number of case reports. The nature of reaction is more how it occurred, or what was the severity, what were the symptoms. So it depends on that. The type of drugs, plausible mechanism. So we can say, okay, this drug can cause secretions, can reduce the secretions in body. So definitely dry mouth, the patient reported dry mouth. It's okay, there is a mechanism, plausible mechanism. So that is possible that the drug has caused dry mouth. Confounding factors. Confounding factors means any other conditions or anything that confounds or confuses this causality. The patient might have underlying condition. Example is macular edema. They report like there is a signal of macular edema shown, like 10 patients had macular edema. Not sure if it is due to this drug or not. But all the 10 patients had a cataract surgery before having the medication. So that is a confounding factor. So not sure this is definitely caused by the drug. There are other underlying conditions. Population exposure, how far the drug has been exposed to the population. Now, decision making, the next step is the decision making following evaluation. Just 3 decisions on the board. And that's the important part, how to make the decision. Either first, close a signal, refute a signal based on available evidence. Refute means we know we have evaluated the signal now, reviewed all the sources, all the literatures, everything, and based we have solid evidence now this is not caused by the drug. We can close that signal. No actions are required. Continue monitoring. Well, we have some evidence this event has been caused by the drug, or maybe by the drug, or not. There are other drugs used at the same time. Oncology is oncological drug is really difficult to make a decision on because most of the time the patients will be on other chemotherapeutic agents and there will be confounding factors there. So a decision sometimes cannot be made whether the event was caused by this particular drug or the other medications the patient was at the time, continuing at the time. So once a decision cannot be made, we still think there is a possible causality. We decide to continue monitoring. Take further action. That's when a signal is verified. The next action is to communicate to the regulators, to the healthcare professionals, to the patients. So what are the actions following confirmation of a signal? Updates to labeling documents, the summary of product characteristics, patient leaflets, revision of the RMP, the risk minimization plans. So there may be revision of that. They add signals, risks, additional risks as identified or potential, or there may be additional risk minimization measures to be taken, produce patient leaflets or educational materials. That's the revision of the RMP. And provision of safety information directly to the healthcare professional. When there are urgent issues, they are directly informed to the HCPs or patients or public. Okay, so that's a complete process of signal detection. The new data is received, data compilation, and then we detect the signal, analyze the signal, validate the signal, and once validated, it is communicated. And this continues because new data comes available all the time. So this process continues. So the message here, all medicines carry a certain level of risk. Now, signal detection and its assessment plays a key role in ensuring the patients receive safe drugs. Benefit risk evolves throughout the life cycle of a drug. As I said in the beginning, this is more important to see that the benefit risk should be always positive. And this signal detection, the signal management, support regulatory and clinical decision making affecting the populations of patients. And finally, for whom? So this is important for doctors, patients, companies, health authorities, etc. So that's the presentation on the signal detection. Thank you very much for listening. Any questions? Sorry, any
Yeah.
questions?
Okay, the presentation is open for the discussion. If someone has question, they can ask.
Ma'am, we have a question in the chat box. There are too many underreporting in India. How to deal with it? Ma'am, should I repeat the question?
Sorry, can you hear me now? Hello?
Yes, ma'am.
Yes, ma'am.
Yes, sorry. I was unable to unmute. At some point my mic got muted. Yes, so underreporting is a problem not only in India, but everywhere. I know in India there may be more underreporting than other countries or other part of the world, mainly because of lack of awareness of pharmacovigilance or lack of establishment. So 10 years or 14 years ago, when I joined pharmacovigilance, honestly speaking, I didn't know what pharmacovigilance was and what an adverse event was, what is reporting was. So it's lack of awareness. The more people are educated on reporting, then underreporting can be prevented. But still, underreporting is something which happens not only in India, it's all over the world.
Okay, ma'am. Ma'am, there's one more question from a delegate. How do we perform casualty assessment for suspected ADRs as a de-challenge, re-challenge cannot be done for suspected ADRs?
So de-challenge can, sorry, I didn't get your question properly. When you say ADR is adverse drug reaction, there is a causality, a possible causality there. I didn't understand your question on the de-challenge and re-challenge. Is that something mentioned here?
Yeah, ma'am, it is in the chat box only.
De-challenge and re-challenge. So de-challenge means if the drug was stopped and the patient is improved. Re-challenge, if the drug was stopped or interrupted and when the drug was then given, again, the adverse reaction has occurred. So based on if a de-challenge is positive, it's not always, doesn't mean there is a causality. But when you see a re-challenge, there is an implication of causality there. That means second time when the patient was given the same drug, the same event was noted again. So that implies a causality there, if there is a re-challenge. I'm not sure if I answered your question right.
Ma'am, the same delegate has one more question. For strengthening the signal, how many case reports are required?
Okay, so for strengthening the signal, as I said, it depends on the company, depends on the product, depends on the number of AEs received per year. Example, if accumulative number of events is less than 1,000, a quantitative method or statistical method may not be accurate. But if it's as a product with lots of events, thousands of events in the database, then statistical method will be a better option. So strengthening for signal, how many cases So it doesn't require number of cases actually. So if the case numbers are less, as I said, qualitative method is a better approach. Looking for designated medical events, reviewing individual cases manually.
Okay, ma'am. Thank you so much. Participants, any other question?
No questions? If you have any questions, I'm happy to answer even via emails. You can reach out to me to my email. Manju should be having my email details and you can pass on to her and provide, I can provide my answers via email as well. You can contact me anytime.
Thank you. Thank you so much, ma'am, for such an engrossing session. Now, everyone is requested to put on their videos for a screenshot. Now we'll have a short tea break and we'll be back by 3.
Kangan, let
Thank
me
you.
thank Madam.
Yes, ma'am, sure.
Yeah, please. Okay. So thanks a lot, Lizzie ma'am. So it was really a very nice presentation given by you, and definitely it is going to help all the delegates which are right now present attending your lecture. And you have talked about signal detection, its importance, benefit risk balance, safe use of drugs, special situation where the signal detection is done, data sources for signals, aggregate data, signal prioritization, signal evaluation, decision making. So these were important points, and definitely it has increased our concept about signal detection in reporting of adverse drug reactions. So thanks a lot, ma'am. On behalf of Pune College of Pharmacy, as well as on behalf of organizing committee, I thanks a lot to you. And then now Kangan can take over. Thanks
Thank
a
you
lot.
very much.
Yeah.
Thanks for giving us the opportunity to speak. Thank you very much,
Thank you.
everyone. Bye.
Thank you so much, ma'am. And we'll just meet after a short break now. We'll end the meeting over here.